Hello everyone. So today we are going to be looking at something I think will be very, very important to share with you guys. In some cases, they usually ask questions about whether you understand how the analyzers work in the laboratory. So they may want to know, do you know the principle of the hematology analyzer? Okay, basically the full blood count analyzer. So they may want to know, do you know, okay, how do they work? Which type of analyzer have you used and what is the principle? And in some cases, they may even want to ask you questions about their own analyzer. But what I'm going to tell you, I'm going to share a general information that is common with all analyzers, and I'm also going to try to mention few that differ from one analyzer to another analyzer, okay? But once you understand the overview, how it functions, then you can always give them the answer and you will never get it wrong going that way. Anyway, once again, my name is Dr. Emmanuel Obodo. I'm a specialist biomedical scientist, and I'm also a lecturer here in the United Kingdom in biomedical science department. Yes, I did put my LinkedIn here. I'm thinking that where possible and you want to have any question, you want me to talk to you, you can always contact me here. Can I also ask that please be patient and watch the video to the end, then you can be able to make use of all the information that has been provided. So yeah, let's get into it. First of all, I want to start off by letting you know that this hematology analyzer measures full blood count. So that is what it measures. And there are three main blood cells: red blood cells, white blood cells, and platelets. So that's what they are measuring. So from the red blood cells, you get red cell indices, and from the white blood cell, you can get total white cell count, and you can also get differential count. And of course, you can also get platelet indices, okay? Now, when it comes to analyzer, I want us to know that there are three main parts or types of this hematology analyzer or full blood count analyzer. In some cases, you can say there are two parts, okay? Because in most cases, some of the the 5-part differential cell count, in most cases, they can also do that of the 7-part. But I decided to group it differently. So I've called it 3 types based on differential white blood cell differential count. So in hematology analyzer, like full blood count analyzer, you have 3 parts: differential cell counter, and you have 3 parts differential cell counter, you have 5 parts cell differential counter, then you have 7 parts differential cell counter. What exactly are they doing? What they are doing is very simple. So there are some analyzers. Remember that differential count has to do with different types of white blood cells. So you are looking at neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So in some cases, there are some that can only give you 3 parts, and they are not able to do the other ones. For an example, they may give you neutrophils, give you lymphocytes, and then give you mixed cells. And that mixed cells will contain the monocytes, eosinophils, and basophils. While the 5 parts are the one that will actually give you the 5 differential count you're looking at, which is neutrophils, lymphocyte, monocyte, eosinophils, and basophils. Then in some cases, like 7 parts, they can then include something like immature granulocytes or maybe blast, as the case may be, okay? So we're going to look at it in detail. So let's look at this. So I thought I should show you this graph, or this table actually, show you the difference between the 3 parts of differential cell counter and 5-part differential cell counter and 7-part differential cell counter. So in most cases, the one that are 3 parts, okay, they use what we call electrical volumetric impedance, okay? That's what they use. And of course, it work on the principle of just the cell size and the volume of the cell, then to determine what is actually in the sample. Then it can also look at the differential white cells. Like I've told you, they are able to differentiate mainly neutrophils, lymphocytes, but they cannot differentiate the rest. So they will call the rest mixed cells, okay, counter. Then what happens is that because of that, obviously they are unable to differentiate between the monocytes, eosinophils, and basophils. So those ones are called 3-part differential cell counter. Now there's 5 parts. Most of the 5 parts, okay, in addition to maybe this very impedance we've used, they can also use flow cytometry, okay, light scatter, where they are able to determine the cells, looking at granularities and diameter and inner complex of the blood cells, okay. Then they are able to differentiate each of the white cells, okay. Then, but when the same thing happens, so these 5 parts and 7 parts, they are almost the same. The only thing different is that when it comes to these 7 parts, there's an addition of maybe, sometimes there can be immature granulocytes, there can be blast cells, there can be atypical lymphocytes, and so on. So with these very 7 parts, which is in most cases the one that is commonly used in the laboratory here in the UK. So these 7 parts will show you both the immature granulocytes, and also blast cells or atypical lymphocytes, as the case may be. So they are called 7 parts. So they are called 7 parts because in addition to the 5 parts of this common differential white cell count, you can also get immature granulocytes and blast cells, or you can get atypical lymphocytes, as the case may be. So that's why they are called 7 parts. I hope that makes sense. Okay, now let's look at the general principle. So in the general principle, before I continue, I'm going to tell you that irrespective of any analyzer that you've used before, okay, if you can't remember everything, try to at least remember the general principle. That is how all of them work. Okay, so first, the analyzer will aspirate the blood. Okay, following the aspiration of the blood, it can then mix with diluent, okay, then and also with other reagents. Okay, those other reagents, generally what most of these reagents does is that it will help to lyse the cells. Once it lyses the cells, it will then expose whatever thing that it wants to measure. For example, following the lysing of the red blood cells, okay, or the blood, it can expose hemoglobin. Then the analyzer is able to measure the hemoglobin. While in some cases it will expose the white cells, okay, and of course the analyzer will then measure the white cells, okay. And in some cases, maybe what it does, that some of these analyzers, what it does is that it kind of make a hole, okay, on the membrane, okay, of any of these various cells, and that way it can now help the reagent, to kind of again expose whatever thing that is in that very cell, and then it can be measured. Anyway, but following in all of this whole thing, so following the aspiration, mixed with the diluent or normal saline, okay, then following that, then with the mixture of the reagent, and that can help, to expose whatever thing that the analyzer wants to measure, whether it is white cells, platelets, or red blood cells, okay, or hemoglobin, as the case may be, then that would then lead to the quantification. So then the analyzer will be able to quantify what is actually in that very sample. That is where it can be able to quantify, hemoglobin there, the white cell count there, and other. Then following the quantification of what is there, it can then classify what is there. So that is where it can be able to say, okay, this is the amount of the white cells, this is the amount of this, and this is the amount of that. And following that very classification, it can then go through electrical or optical techniques, okay, where it can then, through the light, okay, the light will then help to describe the cell population. So you can then be able to know this is neutrophils, this is monocytes, this is eosinophils, this is the basophils, and so on, okay, and this is hemoglobin. So it's from there you are able to be able to, that is how most of them work, okay. Because what I want you to know, that in addition to this, there are more detail of what happened when it comes to the analysis of the full blood count sample, but this is the general principle. Most of the analyzers used in the hematology laboratory, okay, can measure through photometric method for hemoglobin. This one is common for almost all the analyzers, okay, because for hemoglobin measurements, following the aspiration, the red cells are lysed, and once the red cells are lysed, it will just expose the hemoglobin, and that hemoglobin will then be, light will pass through it, and then that can be quantified. That is photometric method for hemoglobin. Then when it comes to the cell counter, okay, you now have electrical impedance, okay, which is actually called Coulter principle. This is where most Beckman Coulter analyzer comes in, okay. Then electrical impedance, that is the principle, while something like Sysmex uses something like flow. In addition to electrical impedance, Sysmex also makes use of flow cytometry by volume conductivity scatter, which is VCS, okay. So basically, when it comes to the analyzers, the main thing that happens is photometric, which is how they measure hemoglobin, and also electrical impedance or flow cytometry, okay. So some analyzers make use of electrical impedance, while some make use of both electrical impedance and flow cytometry. This is how they are able to differentiate the different cells, like different white cell count. Okay. So now let's look at a little bit detail, because I'm trying to give you, gradually, so that you can, at the end, we're going to try to find a way to summarize this. Okay. But let me give you another side of this. So we've known photometric, okay, for hemoglobin. We've known electrical impedance, okay. We've known flow cytometry in terms of differential white cell count. Now, how does this photometric method work? How does it work? It works by what I've already said. The red blood cell, when the sample is aspirated, once the analyzer once it's aspirated, following the aspiration, the red blood cell will be lysed. The lysing of these very red blood cells, okay, would then release the hemoglobin, and that hemoglobin that is released would then be converted to color measurable compound. So once it's converted, then once light passes through it, okay, and that can now be measured, give you absorbance, and that absorbance can then determine what is actually there, the quantity of what is there, or the volume of what is there. So that's why I say monochromatic light is then passed through the solution. Remember, when you go for your interview, you don't need to maybe mention monochromatic. If you think you forget, just remember light will pass through it. Okay. So then, following that light passage on that very light mixture where hemoglobin has been exposed, so light will pass through it. Okay. The passing of that very light, there will be absorbance. Okay. The absorbance from that solution then is measured, by photosensor in the hemoglobin flow cells, and that is how this principle of photometric work when it comes to hemoglobin. Let me take it again. So once the sample is aspirated, following the aspiration, the sample will then be mixed with what? The diluent. Okay. The diluent, once it mixed with the diluent to kind of dilute it, and following the dilution, that will then lead to some other reagents to mention in other video that I'm going to make. Okay. So some of these reagents that will be used, will then lyse the very red cells. Once it lyses that very red cells, that will then expose the hemoglobin, and following the exposure of that very hemoglobin, light will pass through it, and the light passage through that very mixture of that solution would then lead to absorbance, and that absorbance can then tell us, the analyzer will then measure the amount of the hemoglobin in that solution. Okay, so that is what happened with photometric. So let's look at electrical impedance. In electrical impedance, so it uses a counter. It's used to count the white blood cells, red blood cells, and platelets. And how is it able to do that? It's able to do that because there's what we call aperture. So the aperture is just like a hole. So as these very cells, in the whole blood, as they passes through it, okay, it knows, the quantity or the size of the white cells, the size of the red cells, the size of the platelets, okay. As they are passing through these very apertures, okay, there's going to be electrical resistance change. And that electrical resistance change, as the cells passes through the aperture, will generate a voltage pulse, okay. So that voltage pulse that is being generated as the cells passes through that very aperture, okay, which form the electrical resistance change. So the voltage pulse then would then, remember that because that voltage pulse at this point, remember that there are different cells, okay? So these very different cells would then, there's going to be different pulses, meaning as the neutrophils pass, it will give a certain kind of pulse. As red blood cell passes, it gives a certain kind of pulse. As platelet passes, it will give a certain kind of pulse. And once the number of pulses during a cycle correspond to the number of the cell counted. So as the cells passes through, okay, the number of the pulses that goes through shows the cell count, okay. Now, in addition to that, the amplitude of each pulse is directly proportional to the volume, to the cell volume. What does that mean? So remember that the neutrophil will have a different kind of pulse, monocyte will have a different kind of pulse, okay, eosinophil will have a different kind of pulse, and of course, white cells will have a different kind of pulse, and the red blood cell will have a different kind of pulse, and platelet will have a different kind of pulse. So each of these very cells forming these different pulses, each of them having this particular pulse, would then plot as a histogram. Okay, it will now, the analyzer will then plot a histogram. That is where you can say, okay, the red cells, they are normally distributed, or the platelet, they are normally distributed, and so on, because each of these cells, kind of form different pulses, and then that can be measured, as a histogram, and that is how the analyzer will be able to differentiate each of the cells. Okay, so that is how this very electrical impedance work. So let's look at the next one, which is a flow cytometry, okay, by volume conductivity scatter. Okay, so what then happened here is that this is actually mainly common. Remember when we were talking about different parts, like 3 parts and then 5 parts, and I did mention 7 parts as well. So this is, and I did told you guys that I did say that both the 5 parts, in most cases, they also contain 7 parts. So what I said, that when you talk about 5-part cutter, you are saying that this analyzer measures all the different types of the white cells. But when you say 7 parts, what you are looking at, in addition to that 5, they can also give you a typical lymphocyte, blood cells, or a matured granulocyte. Okay, anyway, so this very technique is common in 5/7 part differential. Okay, so it kind of gives more detailed information about the morphology of the white cells. So it doesn't just give you the count of the white cells, it can actually tell you some more information about the white cell. It might give you something like, this is atypical cells, it can query atypical cell, it can query blood cells. So it will give you additional information. If there is immature granulocyte, you will also see it there. It will also give you that information, it will flag it up. So this is why this very technique is one of the reliable, techniques. So it uses both the impedance and flow cytometry. Okay, so impedance and flow cytometry, and like we mentioned previously about the impedance. Okay, so what happened that the cells passes through this aperture, okay, in a single file. So the red cells pass, the platelet pass, the white cell passes. So as each one of them are passing through these very apertures in a single file, there's going to be a lesser beam focusing on each of them. As the lesser beam is focusing on each of them, what then happens that there's going to be absorbance. So that will generate an absorbance, and that absorbance will also lead to a scatter light. And this scatter light that is then generated will then be measured at different angles. So that is why you can see something like lymphocytes is here, you see monocytes, you see neutrophils and basophils and eosinophils. So they'll just be differently. So each of the different angles where you see them is proportional to the lesser beam, the absorbance and the scatter light, and that will then lead to the depicted as scattergram. So meaning that it will show you their different population as a scattergram. You just see this is the amount of this is the amount of that, and this is the amount of this. So I'm going to show you that in detail in the next slide. So let's look at this very differential scattergram. So you are going to see the typical example of what I was trying to mention on the previous slide. So if you look at it, you're going to see that this is a lymphocyte, this is a monocyte. Here you get neutrophils, and if there is basophils, you get basophils. Then here is eosinophils. Now, let me start by saying that there is y-axis, there is x-axis. So this very x-axis commonly measures granulocyte, while the y-axis commonly measures the size of cells. So remember that something like lymphocyte and monocyte, they are agranulocyte. They don't have granules, okay? So that is why you can see them this side, okay? Why, when you compare to something like neutrophils, basophils, and eosinophils that are granulocyte, you can see them at this very particular side. So that's why I said agranulocyte on the left side. So this is the left side where you can see monocytes and lymphocytes because they are granulocytes, okay? While granulocyte is on the right side, this is where you can see them on the right side, okay? Now, remember again that monocyte is larger in size compared with other cells, okay, like lymphocytes and neutrophils and so on. So what happened, that's why it's always at the top, it's always towards the top side here, okay? Lymphocyte will be here, others will be underneath, the monocyte will be a little bit high. Why? Because this y-axis is measuring the size. So that's why you can see it at the top, okay? At the top of the scattergram. Now, having said that, that's why I said that large cells at the top of the scattergram, like the monocyte. But remember, where there is abnormal cells, like blood cells, a typical lymphocyte, they will be larger in size. And because they are going to be larger in size, you're going to see them up here, okay? You're going to see them more at the top, okay? More than that of the monocyte. So what I'm trying to say that once the larger the size of the cells, the more is likely to be at the top. And that's why if you watch my video on the interpretation of the full blood count result, I did say that once you see any cell that start going up, you start seeing a population of cells up here, you need to start suspecting abnormalities. Okay, anyway, so a typical lymphocyte or blood cells is going to be extremely at the top. You see it here. Okay, then what happened at the background here, you're going to see the lower left corner, like here, you're going to see something like nucleated red cells. Also, you're going to see something like platelet clump, if there is any. You see them here. Anyway, so this is how the scatter, gram look like, following the flow cytometry and of course impedance and flow cytometry. So this is actually what I was trying to talk about. Okay, so let's look at fluorescent flow cytometry. This is also another type of measurement. Okay, so but again, this also occur in addition to VCS, which is a volume conductivity scatter. Okay, like flow cytometry, like we've mentioned. So what happened here is that they use it what we call fluorescent dye. So what happened that these dyes will kind of stain the cells, and this will help to give more detailed information about the cells. That's why some analyzers are able to give you they will flag a lot of information. Okay, they may tell you a lot of things. They may say something like there's a nucleated red cells here. They may say something like there's a high reticulocyte count. They may say something like maybe there is platelet clump. They query platelet clump. They query all sorts. So why they are querying that is because of this very fluorescent flow cytometry, where it uses different specific stain dye to stain the cells and thereby help to review that very cells. And following the review of that very cell, it gives the analyzer the opportunity to be able to have more detailed information about each of the cells and flag it up for the biomedical scientists to review it. So this is actually why the advantage it has is that it can also help in analysis of nucleated red blood cells, platelets. In terms of platelets, I mean maybe something like platelets. It can also help to look at something like differentiate matured and immature platelets, and of course it can also help for reticulocyte count. So this is very, very vital, especially in some analyzer like Sysmex. It can be able to tell you maybe there is high retics. It will just flag it up, high reticulocyte count. It can flag it up, high nucleated red cells, platelet clump, and so on. It will tell you all sort of information. This is because of that very fluorescent dye that brings specificity in that. So anyway, that is for fluorescent flow cytometry. Okay, let's look at the video that I want to show you, which will give you idea about the aspiration and the dilution and what happens in terms of the cells passing through the aperture and so on, and how light passes through it for the measurement to be able to occur. Okay. Now, I'm going to try to summarize everything that we've said. So you've gone for an interview and they've asked you, can you tell us the principle of the analyzer you've used? Okay. So what you need to remember is a few things that I've said. Let me start from the very first beginning principle that I gave. I call general principle. What will happen is that no matter any analyzer that you mention, they are going to do this. They will aspirate the blood. The analyzer will aspirate the whole blood, and following the aspiration of the whole blood, it can mix with the normal saline or diluent. Okay, so following the mixture with the diluent, okay, the analyzer will now be mixed with different types of reagents, and that can help to lyse the cells and expose the cells. Okay, where following the exposure of these very cells, okay, there will be light passing through that very cells or mixture, and following that, okay, there's going to be a change, okay, and that will then be measured either by electrical or by optical method. Okay, now the electrical, I mean in terms of when we're talking about electrical impedance, optical method in terms of when we're talking about photometric. Okay, now in addition to that, remember that we now say that is basic. Then we now say that most of these analyzers, they can use photometric, they can use electrical impedance, they can use flow cytometry, scattergram or flow cytometry method. So this is what they do. So what then happens is that each of these analyzers, once they aspirate blood, following the aspiration of the blood, that blood will then be diluted using a diluent. Okay, the diluent then, after it has been mixed and it has been diluted, then it can then be mixed with different reagents, which I said I'm going to talk more in detail in my next video about hematology analyzer. So once it's mixed properly, following that mixture, okay, it can then, in most cases, it will lyse the cells. Remember that this analyzer, they have different channels. You have red blood cell channels, you have retic channels, you have platelet channel, you have white blood cell channels. So each of these channels, okay, so once that is lyses the cells, they will not go to their different channels, okay, and then this test, each of these very reagents can now help to kind of, in some cases, then stain it, and following the stain, and of course, the cells will then be passing through the aperture we mentioned, okay, and I said that the aperture is just like a hole. As it's passing through that very aperture, there's going to be change in the electrical current, thereby leading there's going to be a change in the resistance of the electricity, and that will now lead to what will now lead to pulses. And each of these very pulses will then be measured, okay, and then that can be plotted as a what? Histogram. Then, but when you talk about in terms of the flow cytometry, then what then happens is that as each of the cells are passing through that very aperture again, okay, and each of them are passing as in a single file. As they are passing, there's going to be a lesser beam that will focus on it, and that lesser beam that will focus on it will then generate what is going to generate the lesser beam will then generate an absorbance, and that absorber will then generate a scatter, and that is why each of that very scatter can then be distributed at different angle depending on what that very cell is, whether it is granulocyte or agranulocyte or matured cells or immature cells, as the case may be. So that is the main principle of this. So now this is just the overview of what these very analyzers, what they do. So what I'm going to tell you that be smart about it. If you say you are using a Beckman Coulter, you might be looking at electrical impedance. If you say you are using something like Sysmex or Sysmex analyzer like X and line, you are looking at impedance and electrical flow. You are looking at electrical impedance and flow cytometry, okay, and even fluorescent light. So then you might need to talk about all of them. But if you go for interview and they ask you question on the principle of analyzer, you are looking at photometric, electrical impedance, and flow cytometry. Okay, remember that photometric is for all of them, but some of them, when it comes to differential count, some of them use electrical impedance, while some of them use flow cytometry. I hope this makes sense. I'm not sure if you have any question. If you do have any question, put it in your comment section, and I'm more than happy to discuss this further with you. And of course, you can always contact me. I did put my detail about LinkedIn. If you want to contact me on LinkedIn, you are free to do that. Okay, if you have any question, like I've said before, do ask your question. Please, can I also ask that you subscribe and continue to share with your friends and family. Thank you very much till I come back your way again, and I wish you all the best. Bye.